Ladies and gentlemen, time is on now. We start our faculty colloquium, and today we have a great occasion where two ex aequo scientific prize winners will present their works. The prize is founded by Dr. Marek Pieńkowski. Starting from this year, he is a co-founder. This is a scientific prize in the name of Professor Stefan Pieńkowski, founder of Polish physics. First, I shall call Professor Paweł Kowalczyk, the head of the Institute of Experimental Physics, to say few words about the prize and prize winners. Professor Kowalczyk, floor is yours.
Okay, so good afternoon, everyone. As most of you probably remember, for nearly 20 years now, each year the Professor Stefan Pieńkowski Scientific Prize has been awarded under the auspices of our faculty for achievements of young scientists in experimental works in the field of natural sciences. And up to now, we already had laureates from physics, chemistry, biology, and medicine. The initiator of the idea and founder of the prize was Dr. Marek Pieńkowski through his Marek Maria Pieńkowski Foundation. Last year, a co-founder joined us, namely the well-known Kościuszko Foundation from New York. And this resulted in changes of rules for awarding the prize. So first of all, it has been decided that particular emphasis will be put not only to scientific achievements of the candidates, but also to their ability to form a research group. And consequently, the upper limit for age of the candidates has been increased up to 40, giving them more time to attract coworkers and to formulate their own projects. Then having two sponsors allowed to raise the monetary value of the prize substantially. And finally, the prize starting from this year will be awarded every 2 years instead of every year. Candidates for the prize can be nominated by faculty councils or research councils of Polish universities and research institutes. And this year we had quite a lot of very good candidates. The selection proceeds in 2 stages. First, a pre-selection on the basis of submitted documents, and then auditions with the best candidates. And for the first time in the history of the prize, we were not able to select a single winner, and finally decided that the prize should be shared equally between 2 persons, namely Dr. Anna Karnkowska from the Faculty of Biology of the University of Warsaw and Dr. Dorota Skowron from the Astronomical Observatory of the same university, which is actually a part of our Faculty of Physics. The final requirement to receive the prize is to deliver a lecture at the colloquium of the Faculty of Physics. This is a bit like the idea of a Nobel Prize lecture. And therefore we shall have today not 1, but 2 slightly shorter lectures, one on evolution of very simple one-cell organisms and the other on construction of a map of our galaxy. The subjects are very different, but I hope that both lectures will turn out fascinating. Thank you.
Thank you very much. Do we have Dr. Marek Pieńkowski with us? He is the founder of the prize, and we expected him to get connected to the colloquium today. No. All right. So I would well, we have to skip his introduction. So I shall ask Mr. Marek Skulimowski, the president of the Kościuszko Foundation, to speak to us. Please, the floor is yours.
Good afternoon in Poland, and thank you so much for inviting to this ceremony. And first and foremost, let me congratulate to the winners of this year award for their achievements, of course. And I hope it's going to be a sort of a beginning of their wonderful career with a lot of similar and even bigger in monetary prize awards in the future. And I hope I'll meet you one day at the Kosciuszko Foundation. You can see that in the background. Of course, it's a fake background, but at least the photo is of the Kosciuszko Foundation interior. And I would also like to thank the committee for the hard work on selecting these wonderful candidates, and the founder of the award, Dr. Marek Pinkowski, who is also the Kosciuszko Foundation trustee. So again, let me congratulate and wishing you all the best in your careers and for the coming holiday season. Thank you.
Thank you very much. Before I call for the first speaker, a few comments or organizational comments. Please, we shall organize it in such a way that each person will have 30 minutes for their lectures. And then after each speaker, we shall have 5 minutes for questions, which please mark on the chat. I shall be watching the chat and seeing how many questions and who should ask them. Also, there is a kind request to those of you, to those participants who don't sign with name and surname, to do so, because Zoom is keeping a record who is in the audience. And as you remember on this colloquium, we used for ages, this ages-long tradition, we sign a book just for future generations. So please rename your name into a proper first and second name, so we keep a record who was present. Please do so. If you click on participants and then at the side of your name you have something like more. If you click on more, then you can rename and well, be recognizable by name and surname. Thank you very much. So now I shall call Dr. Anna Karnkowska from the biology department to teach us and lecture to us about exploring microbiological diversity to understand the evolution of eukaryotic cells. Please, Anna.
Thank you very much. I'm going to share now my presentation. Is it visible?
Yeah, it's perfect.
Yes. Okay, great. Thank you very much for the introduction. I'm really honored by, of course, by the award, and I'm very honored also to speak at this colloquium, which, as I expect, audience is quite different than I'm used to talk. So keeping this in mind, I tried to prepare this for non-biologists. So everybody who's biologist on the audience, and they are as well. I'm sorry if some things will be really simplified, but I hope that that way I will be able to share with you why I'm interested in this topic, and I will try to tell you today about how and why we explore the microbial diversity and what we can learn from this diversity about the evolution of life on Earth. So let's start from moving to some place where we expect high biodiversity. Of course, biodiversity actually doesn't have to be high in this kind of places, but since today is really gray and dark outside, I thought it will be nice to start somewhere where it's bright, sunny, and green. And if we would happen to land on this kind of island, we might see some organisms. And those most likely will be plants and animals. And even if you're not a biologist, you would be able to recognize that this is an animal and a plant. And if we take a walk on this island, probably we'll find other species as well. And even if you don't know the names, you will know that this is something different. So that's actually diversity, right? And that's how we see diversity with our eyes. And we don't have to be really well trained to do that. And that's how we have seen diversity from most of our history. And in 18th century, Linnaeus put this into some frames, and he named one of these groups animals and the other plants, and we use it now as well, of course. And those were all multicellular organisms. However, with the development of the microscopy, we started to learn that there are actually a lot of other forms as well. And those are microscopic, and we only can see them with some equipment. Some of them are bacteria, some other are protists, and I will talk about them a little bit more later. But it basically turned out that the diversity is much more complicated than only plants and animals if we have some equipment to see that. And then in 20th century, in mid-20th century, where the structure of DNA was discovered and subsequent technologies developed to sequence this DNA and to which enable us to read the genomes and genes, we learn even more about diversity. And quite early already, in just after a few years after the DNA structure was discovered, it turned out, for example, that fungi, which were usually thought as plants or were treated as plants because they are not moving, actually, based on their genetic information, we know that fungi are more closely related with animals than with plants. So all these discoveries led us to some deeper understanding of the diversity of life. And some of these forms are big, right? We can see them with a naked eye. Those are multicellular organisms such as plants, animals, some fungi. And some are unicellular and very small, so we only can see them under microscope, such as protists and bacteria. So that's why one way of looking at this diversity. Another way, if we still keep in mind the same simplified view of diversity, is to look on how those organisms, their cells, look like. So what's the structure of their cells, single cells? In case of bacteria, which are one of the best known representatives of group, which we call prokaryotes, we have different cells than the cells which are in those other organisms, such as animals or plants or protists, which we call eukaryotic cells. The main difference, to simplify it, is that eukaryotic cell has organelles, including most importantly nucleus, where the DNA is closed, enclosed, or other organelles such as mitochondria or chloroplast, and many others. So despite the fact that if something is unicellular or multicellular, actually we can take a look also on their structure. And then the unicellular forms such as bacteria are prokaryotes, and bacteria are only unicellular, but the eukaryotic cells might be unicellular, such as protists, but also other organisms might have the same types of cell, eukaryotic cell, even though they're multicellular. So in principle, our cells are eukaryotic cells, and also some amoeba will be eukaryotic cells. So our basic structure of our cell is very similar. And now if we take these techniques, these microscopy techniques, model techniques, and sequencing techniques back to this island, and we'll explore the diversity of eukaryotes, I will be focusing now, from now on, in majority on eukaryotes. If we take a closer look on the diversity using these other techniques, not only our eye, it will turn out that actually majority of this diversity is a microbial diversity. Obviously, bacteria will be really diversified there, but since I'm focusing today on eukaryotes, I just want to show more or less the percentage of, or the dominance of microbial eukaryotic diversity over multicellular eukaryotic diversity. So the plants and animals which will be visible there are only a small portion of the whole diversity, whereas the other diversity is hidden for a naked eye, and we can take a look on that using microscopy or sequencing technologies, and that's actually the majority of the diversity. What I mean by this diversity of microbial eukaryotes, the other name of this group are protists. So I'll be using both these names. You might know from school also the name protozoa, which is not exactly the same, but it might be the word you know for that. So those are all these unicellular eukaryotic forms, which are morphologically diversified, as you see here under microscope. They might differ in lifestyle. There might be parasites, there might be predators, they might be photosynthesizing organisms, and they might live in different environments, of course. They will live in the water, in the soil, in the intestines of other animals. But more importantly, they are really diversified at the level of their genomes. So they are distant from each other. So that's a simplified view of eukaryotic diversity. Here we have in gray, all gray branches are eukaryotic lineages of unicellular eukaryotes, and the green, purple, and blue line are those which are multicellular. So plants, fungi, multicellular fungi, and animals are only a minority of these different lineages, whereas the diversity, majority of this diversity, genetic diversity, is hidden in these gray lineages, which are unicellular. Moreover, the multicellular organisms actually evolved from these unicellular ones. So to understand their origin, their evolution, we have to study unicellular ones. And also, if we try to understand the origin of all those organisms, we try to find the organisms which will be most similar, most close related to the common ancestor of all of them. And this common ancestor was most likely also unicellular. So the rest of my talk, I will be focusing a lot on the cell biology, and I will simplify the cell biology, but I know that we might have a different view of the cell biology and what is inside the cell. So do not worry too much that I might have quite complicated view of that, and your view might be a little bit different. That's not really the most important, because what's important for me here is that we'll see that this cell is complex. We don't have to know exactly what each part is doing to see that it's really complex. And there are a lot of things in the meaning of the organelles inside. And I'm interested in how this complexity evolved. Of course, eukaryotic cells also do not like exactly the same, all of them, but they're all really complex, or much more complex than prokaryotic cells, from which they evolved. So the question is how this all evolved in the meantime, and how that happened, and what are the origins of all of these organelles. I'm not working, of course, on all these problems, but we cannot skip this very early stages to understand even the later stages. So one of the major questions in the evolutionary cell biology, so evolutionary biology of eukaryotes, is how basically this eukaryotic cell emerged. And one of the most important parts of this cell is a nucleus, which I mentioned. Inside of the nucleus, there is a DNA. So that's a feature of eukaryotic cells. So we would like to know when and how this emerged. The estimation is that it was around 1.5 billion years ago. And this cell evolved from proto-eukaryotes, so some sort of a prokaryote actually, but this major step as emerging of the nucleus, this is not very clear how it happened. There are various hypotheses, and among other problems, there is a question if actually nucleus came first, or the other organelle, which is called mitochondrion. We know a little bit more about how mitochondrion was created or emerged. And here I just want to come back for a second to the term of a symbiosis, which I hope is quite familiar for you, but just to remind, it's in simple words, just an interaction between two organisms where both of them are somehow mutually beneficial from that interaction. Here we have a fish inside of the sea anemone, which is a shelter for those fish, and they clean it from organic matter. So this is a general concept of symbiosis, very simplified. And we also there are other types of symbiosis, which we call endosymbiosis, where just one organism is inside the other. And that might happen also at the level of single cells, of course. So that's how we now interpret the origin of mitochondria and also actually chloroplasts. So the evidence is that they evolved through the endosymbiosis. So this kind of a close relation of two organisms. In this case, the host was some sort of a proto-eukaryotic cell. As I mentioned, we are not sure exactly if that was already with fully developed nucleus or maybe not. That's one of the questions. But that was definitely some ancestor of eukaryotes. And this host had an interaction with alpha protobacterium, and that gave rise to mitochondrion. That single event happened, at least successfully, only once. So all mitochondria originates from single ancestor, and they all are related to each other, therefore. So it happened early in the evolution of eukaryotes, and then all eukaryotes just have their mitochondria coming from this single event. That's also happened similar way with chloroplasts. And of course, not all eukaryotes have chloroplasts, only those, well, basically higher plants and their relatives. And that was also an event, endosymbiotic event. In this case, already established eukaryotic cell was a host, and then the endosymbiont was a photosynthetic bacteria from the group cyanobacteria, which gave rise to chloroplasts. So those are very unique events, which happens very rarely in the evolution, but they turn the direction of the evolution. So those two organelles, endosymbiotic organelles, I'm claiming that they're really crucial for what we can observe now around us. Why? Because they're both involved in energy metabolism. I will be focusing more today on mitochondria, although I also work on chloroplasts, and we can compare each of them to certain energetic, certain energetic purpose. They serve certain energetic purposes. So mitochondrion is something more or less, we will call it powerhouse for the cell, and the chloroplast is rather like a solar panel, right? But both of them are very important. Thanks to the chloroplast, obviously, we have all the photosynthetic eukaryotic organisms. So higher plants on this island would not exist if not the origin of chloroplast. But mitochondria actually were more important in the early stages of the evolution. That is because thanks to the mitochondria, these cells which obtain mitochondria, early eukaryotic cells, were more effective in energy generation than prokaryotes. And the cells start to grow, might grow larger, and they have enough energy to also their genomes grow larger. I mean, they duplicated, the genes duplicated. So there was enough material to diversify. And that helped help in early stages in a very weak diversification of eukaryotes. And later on, that led, of course, to these many lineages, which I showed you on this tree, but also from them, of course, evolved multicellular organisms. So that's a central role of mitochondria in the evolution of early evolution of eukaryotes. What's important here is that mitochondria actually are not only taking part in this process, which is energy generation in an aerobic environment, but also because they became really important part of the cell, and they cannot be separated right now, of course, from their host, from the eukaryotic cell. They started to take part in various metabolic processes. Those processes are just, I listed those which are most important. Those are core metabolic processes of the cell, and mitochondria are involved in them. So they're indispensable for the cell because they already taking part in various processes which are important for survival. Those are core metabolic processes and also the synthesis of various metabolites. So I'm interested in two aspects in the evolution of this endosymbiotic organelle, mitochondrion. The first stages, so the origin of mitochondria, it looks very simple on this drawing, on this graph, because it's just endosymbiont which becoming mitochondrion. But this is actually quite complicated, and we still don't know a lot of details. How exactly looked the ancestor of the eukaryotic cell, which alpha protobacterium took part, and how actually this bacterium turned into the mitochondrion, which is now so tightly, so important part of the cell and taking part in so many processes. On the other hand, already established mitochondrion still, of course, is evolving. And thanks to the research on the diversity of eukaryotes, now we know, and we're knowing much more every year about the diversity of mitochondria, their ultrastructure, their functions, and their role in particular cells. So I'm studying, by studying diversity of eukaryotes, actually I'm trying to focus both on this diversity and evolution of already established mitochondria, but through that also shed a little bit light on the origin of mitochondria. So the typical mitochondria, which we can find, for example, in our cells, you might recall that they have their own DNA, they have two membranes surrounding this organelle, and they also, of course, serve a lot of these functions I already mentioned. So they, most importantly, are involved in the energy generation for the process which is called oxidative phosphorylation. And then also they're involved in very many other processes in the cell. But it turned out that eukaryotes actually might also live in anaerobic environments. And that was quite surprising when it was discovered, of course, quite long time ago already, because mitochondria, they use oxygen for this process of oxidative phosphorylation. So how then these organisms live, how they evolved to adapt to these environments? Well, the first anaerobic eukaryotes which were discovered were actually quite important parasites of human, and they might live, for example, in our intestines. So they were very quickly and deeply investigated. And it turned out that they still have their own mitochondria actually, but they are very different and they are simplified and their metabolism is slightly changed. And now we know that this type of anaerobic the anaerobic eukaryotes actually are present in many different lineages, and the switch to the anaerobic lifestyle was pretty common. And then their organelles are simplified, as I mentioned, but they still mitochondria. They evolved from mitochondria. We call them mitochondria-related organelles because they are not anymore typical mitochondria, but they evolved from mitochondria. They do not have their own DNA most of the time. They don't have other structural features, but they still have two membranes, and they might be involved in some of these processes. Most of the time, they're not involved in the oxidative phosphorylation, as I mentioned, because they do not have oxygen around, but so this is lost most of the time, and they might be not involved in any energy generation, actually. And they might also lose some or all, almost all other processes, but they always keep one, which is called iron sulfur cluster synthesis. This was a little bit surprising because this is not the most famous function of mitochondria, but actually all types of mitochondria keep this process. Those which are in typical one and those which are reduced, they always perform this iron sulfur cluster formation in their mitochondria. This is just a simple process operated by one metabolic pathway, which is called ISC, where this pathway put together irons and sulfurs, and then these clusters are transported out of the mitochondria, and they're an important part of some proteins, which are critical for the survival of cell. So no cell can live without that. And with that knowledge, I started to work on this organism on the right, which is called Monosarcomonoides. And this is a unicellular eukaryote, as you might see, isolated from chinchilla, intestines of chinchilla. So it lives in the, let's say, anaerobic environment, and is closely related with other anaerobic eukaryotes, which has already reduced mitochondrial organelle. So my task was to basically describe the cell biology of this organism and also its mitochondria through genome sequencing. That was 5 years ago, maybe a little bit more when we started this project. So the technology already changed, but the main pipeline is similar. So we sequenced the genome, then assembled the genome and made a lot of bioinformatic analysis to understand the cell biology through cell metabolism and cell systems of this cell. That sounds pretty straightforward, This process, which is depicted here, that's took us 2 years, 10 people to work on that, just because it's quite difficult to make sense of all this genetic information. The genome of this organism, which is not the biggest genome possible, is 75 million base pairs, which means 75 million letters, basically, and that's 25,000 pages. And then from that, we have to understand what genes are there, and those are more than 16,000 genes in this case, and what they do, and how they interact with each other, and how the metabolism look like, and what exactly this cell is doing. So that took a while, but in the end, we showed that this cell is, in most aspects, quite typical complex eukaryotic cell. And at this point, we could start to look at their mitochondria. We expected, as I mentioned, quite reduced mitochondria because of the anaerobic lifestyle. On the other hand, we didn't have microscopic evidence for mitochondria in this organism, but that's not very rare for those reduced mitochondria, because they are often really small and they don't have very distinct features. So I just want to show you briefly how we did it. Just 2 examples, how we search for proteins, so some hallmarks of mitochondria. Well, one of the most important hallmarks of mitochondria are the proteins which enable other proteins to go through these membranes, which I mentioned earlier. So any protein which can go inside mitochondria or will go outside has to go through certain complex of proteins, which is really big, well conserved, and it's present in all other mitochondria. So it might lose some of the proteins, but most of them are present and they're well conserved, which means it's relatively easy to identify something similar. So that's what we did. And to our surprise, we did not identify any of those proteins. That was the moment which is quite disturbing for every scientist. If you cannot find something, you obviously doubt that you did it correctly. So because I was mainly responsible for that, so I was searching over and over various ways and thinking how we will prove then, if this is not there, if this system is not present there, that would mean maybe that the mitochondrion is not there, but how would we prove it that it's absent? So what I thought was to search for any other possible mitochondrial process, which we know from mitochondria, and try to identify how this cell is doing this, how it's fulfilling this metabolism, if this pathway, mitochondrial pathway, is not present. So pathway by pathway, I was analyzing those mitochondrial metabolism, and some of these pathways I found just analogous pathways in the cytosol, but for some are really absent, but we could easily explain the absence of them by the fact that the cell lives in the rich environment of the intestine, so it can actually transport some of the metabolism from the host, and we found transporters for that. So some proteins which will help to transport those specific metabolites inside the cell. So with that, basically, I was able to rule out almost all of the important metabolic pathways in mitochondria. And as the last one, I focus obviously on this one of iron sulfur cluster formation, which I mentioned earlier, that this is a one which is critical for all eukaryotic cells. Just a little bit larger view, how it looks in the cell. So this pathway is in mitochondrion. It actually originated from alpha proteobacteria and produced these clusters, and then they're either used in mitochondria, some of them, or they are transported outside, where so to the cytosol of the cell, where another strictly eukaryotic pathway, CIA, that's the name of the pathway, take them and put together with the proteins. And that way, the cytosolic iron sulfur proteins are produced, those proteins which are critical for the cell survival.
You have 3 more minutes.
Yes. So we focus on searching those, and even if the mitochondria is very reduced, it should be there, but we did not identify actually ISC pathway. we found CIA pathway, which would mean that still these proteins are produced. So it was a little bit confusing, but quite easily we actually, surprisingly for us, identified a different pathway, which is called SUF pathway, which is typical for some bacteria. It's usually not present in eukaryotes, not in their mitochondria, and never was identified as a main iron sulfur cluster formation pathway in eukaryotes, but we found this in this organism instead of IC pathway, and we suggested it functionally replaced it, and therefore the remnant, this mitochondrion, remnant of the mitochondrion could be lost. So that's how we suggest it happen, that this SUF pathway, as I mentioned, is not typically in eukaryotes, but in some bacteria. And we proved by other analyses that it was transferred from bacteria to eukaryotes early in the evolution of this lineage, and led to the replacement, functional replacement of typical eukaryotic pathway, ISC pathway, by different one. And that in the end resulted in the loss of mitochondrion in this lineage. Here I just want to mention one thing. That's I know it's a pretty complicated slide, but what is visible here is that's a process and not one particular change. So to really lose a mitochondrion, which happened only once so far, at least we know only one group which lost mitochondria, it's not a single event, it's a process from gradual reduction of various functions. And the critical turning point was this lateral gene transfer of the genes from bacteria to eukaryote, which functionally replaced one of the metabolic pathways, the last one which was preserved in this mitochondria, and eventually led to the loss of mitochondria. And to sum up, that's how the eukaryote can actually, it was possible in this lineage to lose mitochondrion, which happened, as I mentioned, once, and that's how the endosymbiosis can be undone. My take-home message from today depends on probably on your interest in that are quite different, but first of all, I would like, and I hope all of you will remember that eukaryotic cells usually have mitochondria, and there is this one single lineage which have lost them, but in principle that mitochondria are typical eukaryotic features. Then the study of monocercomonoides for evolutionary biology has quite huge implications, because this is a complex eukaryotic cell, as I mentioned. And then if we have a complex eukaryotic cell with no mitochondrion, that suggests that or can shed a light on the early evolution, suggesting that this early eukaryotes might also have been already quite complex before they acquired mitochondria. It is also, for cell biologists, possible interesting model to study all these processes which are taking part in mitochondria and other parts of the cell, because having this cell without mitochondria, we can easily disentangle effects of mutations of various interventions, experimental interventions on mitochondria from other cellular compartments. And finally, I hope that I convince you that studying eukaryotic microbial diversity can really help us to understand evolution of eukaryotes, and therefore our evolution and evolution of life on Earth. Thank you very much. I just have shown some small part of my research, which I started during my first postdoc. So I just here would like to thank all my colleagues throughout my first and second postdoc, and my current team in Warsaw, with which I'm trying to continue work on the evolution of eukaryotes and also ecology and diversity. Thank you very much.
Thanks to you for this very interesting talk and for fantastic timing. You know, you arrived to the end like a Swiss train. And now we have 5 minutes for questions. And now I have Krzysztof M. He just did not want to tell us who he is, but Krzysztof M., please ask your question.
Meissner. Meissner, yes, I have put the name. So I will have the question, what is the origin of microtubules that are very interesting from the physical point of view, because they are suggested to carry some genuinely quantum mechanical information in the cell. Do they also come from endosymbiosis or not?
No. I cannot tell, I'm not able to tell in details how they originated, but endosymbiotic origin right now we believe it's only in case of mitochondria and chloroplast, or other structures in the eukaryotic cell were either in some form of or other were already evolved in the ancestor of eukaryotes. Like the microtubules which are present in eukaryotes obviously were not the same. Microtubules are not present in other organisms, but some proteins involved and some mechanisms probably evolved earlier, and then this started to look like that later in the evolution. I'm not specialist to tell exactly how they evolved, but definitely not, or at least we don't think it evolved through endosymbiosis.
But they are not present in bacteria.
Not in the form, not in bacteria, and not in this form, but archaea actually, which are archaea, which I didn't mention not to blur the view, but archaea, which are other group of prokaryotes, and probably are actually the ancestors of eukaryotes more than bacteria. They have already, I'm not saying that they have microtubules, but they have a lot of proteins which are also present in eukaryotes, and they might use it different way. We don't have also the full view of archaea yet, because the diversity of archaea is another interesting, very interesting aspect. But then some compounds, some bricks, evolutionary bricks might be already there, but they just will not look exactly like that. But I cannot, I never thought about that in detail, so I cannot really answer in precise.
Thank you for a very nice talk. Thank you.
Okay, thank you very much. More questions? Maybe somebody wants to add something or comment? No? All right, thank you very, very much again. And we really wish to soon listen to your lecture on the Nobel celebration.
It's a long shot, but still, thank you.
Time is passing very quickly. All right, let me ask the second exequo winner, Dr. D.Skowron from Astronomical Observatory, who will tell us about a three-dimensional map of the Milky Way. The map is based on classical Cepheids. Please, the floor is yours and you can share your slides.
Thank you very much. First of all, I would like to thank for the prize. I'm very honored. And I will proceed to share my presentation.
All right, we see them.
Okay, that's it. Just a short question. Do you see my pointer?
Yeah, we do.
Okay, so that may easy some showing. Okay, so I would like to tell you about, as introduced, about the three-dimensional map of the Milky Way that we created recently using a type of variable stars called classical Cepheids. It is really difficult to map our own galaxy because we live within the galactic disk. And as you see on this beautiful image of the Milky Way, galactic disk is full of, not only full of stars, but also full of gas and dust that's obscuring our view. We know that we live in a spiral galaxy, that is for sure. We know it also from observations of other galaxies in the universe, such as these pretty images here. But we also know it from our own observations, for example, from observations of neutral hydrogen and star counts along the line of sight. But these observations that lead us to some image of our Milky Way are indirect. That means that they are model dependent. Depending on the assumptions we make, we get a different picture. So there is still no consensus on how exactly our disk looks. And there are still debates on the number of spiral arms. Some studies say there are 2, some studies say there are 4 spiral arms. There's still no agreement on how large the disk is, or what's its shape, and how tightly the arms are wound around the Milky Way. And that is because the methods are based on some assumptions. Now, there are some objects, there is a class of objects that can be used for direct distance determinations. And given a large sample of these objects distributed across the entire galaxy, we could map the galactic disk with great accuracy without any underlying models. And this group of stars are called classical Cepheids. And these are very bright and young supergiant stars. And the closest classical Cepheid to us is Polaris, which you all know. And here on this image, there is a comparison to the Sun, which is here just below Polaris. To give you some numbers, classical Cepheids are very young. They are younger than about 400 million years. They are very massive, 4 to 20 times more massive than our Sun. They are very luminous, 100 to 10,000 times more luminous than the Sun. And their pulsation periods are fairly long. They're from about 1 to about 100 days. And I already mentioned about pulsations, because classical Cepheids are very well pulsating stars. Pulsation means that they change their size and temperature periodically. And what you see on the left is the plot of brightness of the star, how the brightness changes with time. We call it the light curve of the star. And in the case of classical Cepheids, the brightness increases quickly and falls slowly. So why are classical Cepheids good for mapping our galaxy? The first and the main reason is that they obey a period luminosity relation. And this is very important. This allows for the direct distance measurement to the classical Cepheid, and this makes classical Cepheids the so-called standard candles, which you may have heard of. And the period luminosity relation, this is an example. This is a sample of classical Cepheids from the Large Magellanic Cloud. And here on the x-axis, we have a logarithm of period, and on the y-axis, there is the brightness of stars, which we represent as in magnitudes. We say that instead of brightness, we say magnitude. So with different colors, we have different pulsation modes, fundamental mode, first overtone, second overtone, and so on. But what I would like to show here is that the relation is very well defined and it is very tight. And this is a very simple relation between the magnitude, which is the large M here, and the logarithm of period. And it's, of course, wavelength dependent. Now, how do we calculate this distance? We have what we have from observations. We have the period. Period is very well calculated given that we have a sufficient number of observations. That is that the light curve of the star is well covered with observations. As I said, classical Cepheids pulsate with periods up to 100 days, but the precision of period calculation is even less than a second. Then we have the observed magnitude, the small m, something we see in a telescope. And then we also need, this is the most difficult part, we need then information about the interstellar extinction. Extinction is something that scatters and absorbs light between us and the observed object. So this is all the gas and dust between us and the star that makes the star look dimmer. So we calculate the absolute magnitude from period based on the period luminosity relation. And then in a very straightforward way, given the observed and the absolute magnitude and the extinction, we get the distance. So the prescription is very easy. Now, why else are Cepheids good for mapping the galaxy? They are very bright. They can be seen in other galaxies. And in our galaxy, they are seen even through the clouds of gas and dust. They are so bright. And then the last, they are easy to separate from other variables. So if we have a couple of years of observations and a couple hundred data points per star, we can definitely say whether it is a classical Cepheid or not. Here you have examples of classical Cepheid light curves depending on the brightness on the y-axis and on the pulsation period, which is not crucial here, but it's noted on the right-hand side. So, as I said, the prescription is easy, but there is this big uncertainty, which is A lambda. This is the interstellar extinction, which is wavelength dependent. And in the optical bands, it is very, very high. You can see a galaxy, a beautiful galaxy, but the majority of what we see here is the interstellar medium, which is the absorbing gas and dust. And just to give you an idea, this is something which we see through our telescope. This is an image of a sky in the direction of the galaxy center, so very dense region stars. And we see lots of stars in this area, but there are also dark patches on this image. And this is not that there are no stars here, it's just that the light from these stars doesn't reach us at all. So how do we tackle the problem of the interstellar extinction? We go into mid-infrared data. So in the mid-infrared regime, the extinction is very low. So we use the data from Space Telescope that observe on the mid-infrared, that is Spitzer and Wise, and we use the infrared period luminosity relations to minimize the effect of extinction, to picture how the extinction depends on wavelength. This is the plot of the extinction curve, where on the x-axis there is the wavelength, and on the y-axis there is the relative extinction. And mid-infrared stars beyond 3 microns. You see that the extinction curve is almost flat here, while our optical observations are somewhere beyond the scale, probably here. Given the logarithmic scale, this gives 10 to 20 times larger extinction in the optical than in the mid-infrared. So, and there are available extinction maps that provide information about the extinction value in the infrared bands, so that facilitates even further the calculation of the distance. Now, distance and extinction are correlated. That means that the distance calculation is an iterative process. We start with calculate separate distances to each object for every available mid-infrared band. If we take 4 bands from Spitzer and Wise, we get 8 independently calculated distances d lambda. And initially we assume there is no extinction. Then we calculate the mean weighted average, the weighted average distance d of all the 8 or less, if we didn't have observations, individual distances with 3 sigma outlier rejection. And based on that distance, we extract the value of extinction A from the infrared extinction map for that given distance. And then we go back to step 1, and using that extinction, we recalculate distances and their mean, and then extract the new value of extinction, and so on until we converge. Now, the recipe is pretty straightforward. The biggest problem is the Cepheid sample. Thanks to the OGLE project, which telescope you see here is the OGLE telescope located in the Las Campanas Observatory in Chile, and this is the Polish Warsaw University telescope. So thanks to the observations carried out by the OGLE project, we are able to find the Cepheids in our Milky Way. Because the project of observing the Milky Way lasted over 6 years, we found practically all Cepheids that could be found in this area. And given the high quality of observations, we are sure that the sample is practically 100% pure, that this is not contaminated by other types of variables. And here with yellow dots, I overplotted the classical Cepheids that we were able to locate in our Milky Way. Now, this is the view in the galactic coordinates. The yellow dots are the OGLE Cepheids. The blue dots are Cepheids from other surveys in regions unavailable to OGLE. This is, for example, the northern hemisphere or the very bright objects which saturate in the OGLE images. And altogether, we were able to collect over 2,400 of classical Cepheids for our analysis. Now, when we calculated distances, as I shown before, for over 2,200 Cepheids, we were able to get their distances. For these many Cepheids, we had the infrared data available in the Spitzer-Wise archives. And we plotted on top of a schematic image of a spiral structure. This is not our Milky Way. This is just a spiral structure to focus on the location of the sun and the galactic center. So the yellow dot is the sun. The galactic center, of course, is the brightest part. And the green dots are the Cepheids. What we see straight away is that the Cepheids are not distributed randomly. They are found in structures that resemble the spiral arms, although they are not a clear image of the spiral arms. Having their distribution in 2 dimensions, we decided to now to see what it looks in 3 dimensions. And this small image here is the same image as before. It's just that we divided the galaxy like a pizza in those pizza slices, each with a different color. And now for each of that slice, we will see the plot of how the height of the Cepheid above or below the galactic plane changes with the distance from the galactic center. So on the x-axis, we have the distance from the galactic center. And on the y-axis, we have the vertical distance of a star from the galactic plane. We see that in some regions, the Cepheids are located to the south and some to the north of the galactic plane. And if we fit a 3-dimensional, if we fit a surface to this distribution, we see that that the warping of the disk, of the galactic disk, is fairly significant. Please note that the z-axis here is expanded with respect to the x and y, so this is slightly exaggerated to show the extent, but the warping of the disk is significant. And this nice animation shows how it looks from different viewing angles. So the yellow dot is the sun, and the points are the classical Cepheids. In the vicinity of the sun, the disk is warped to the south, and on the other side of the galaxy, it's warped north. And we also note that the scale on the z-axis changes dynamically as the animation progresses. If we were able to fly away and look at our galaxy from the side, this is more or less what it would look like. As the sun again is marked with a yellow dot, and green points are the Cepheids. We see that the galactic disk is not only warped, but it's also flared, which means that its thickness increases with the distance from the center of the galaxy. And if we plot the distance from the galactic center from the vertical distance from the plane after subtracting the warp model, we see that the flaring here is significant. And we also, based on this, we are able to calculate the distance of the sun above the plane of the galaxy and this scale height. So the distance of the sun beyond, above the midplane of the galaxy is 14 parsecs, which is about 50 light years. Now, maybe a summary of the Milky Way disk in numbers. I decided to write it in light years rather than parsecs. The warping of the galactic disk starts at about 25,000 light years from the galactic center, and until then it's flat. Stars in the outer disk are up to 4,500 light years displaced from the galactic plane, and the thickness is not constant. It's about 500 light years near the sun and over 3,000 light years near the edges. And as I said, the sun is displaced by about 50 light years from the midplane of the galaxy. Now, the second thing that we looked at, that was the three-dimensional distribution, but now the physical properties of classical Cepheids. They, as I said, they are pulsating stars, and we noticed that their pulsation period changes, their mean pulsation period changes with the distance from the galactic center. Here on the x-axis, we have, again, the distance from the center of the galaxy, and on the y-axis, we have the logarithm of period. We see that the further, the shorter the periods. And Cepheids obey the period-age relation, which means that older Cepheids pulsate faster than the younger. So the period distribution suggests that there is an age distribution in the galaxy. So we have here we have the same plot as before, the face-on view of the galaxy with the Cepheids plotted with colored dots. And here the color denotes the age of the Cepheid. So on the bottom histogram here is the legend to that plot. So younger Cepheids are the purplish colors, middle-aged are the bluish, and then the greenish yellowish colors are the oldest one. We decided, so we see here that Cepheids that form structures are at the same time of a similar color. That means that Cepheids in structures are of similar age. That the structures, that means that the structures must have been created more or less at the same time, and that in the past there were some star formation episodes that the star didn't. That the stars weren't born continuously over time, but that they were formed in bursts. And to verify this hypothesis, we separated this sample into 3 age groups. And here on the right, you have these age groups. The youngest one on top, the slightly older, and then the oldest ones. And indeed, we do see that stars of the same age form structures. But we decided to run a simulation, and this is a slightly busy plot, but I will try to go over it slowly. Here on the left, in the left column, there is the observed distribution of Cepheids, similar in the previous slide. That is, each row is for the different age bin. So the top row is the youngest, the middle row is the middle age, and the bottom row is the oldest Cepheids. And spiral arms are marked with different colors. So we have the current, let's focus maybe on the top row for now. The current distribution of Cepheids is presented here. We then postulate that 64 million years ago, there were star formation bursts in 3 spiral arms in the galaxy. 64 million is because this is the median age of Cepheids in this age group. So, and as you will see, the galaxy is rotated backwards by, the spiral structure here is rotated backwards by about a quarter of a circle, because the rotation cycle of a galaxy is about 250 million years. So the sun now is here, but then 64 million years ago, it was probably somewhere here. And we do the same for each age bin. It's just that the amount of years that we go back in time is different for each age bin. And then we run this simulation to the current times. So we see what happens after 64 million years here, after 130 million years here, and about after 175 million years here. And this is the result. So this simulation progressed forward gives this result. And if we compare this simulation result here with the observed distribution here, and in the next two rows, we have a surprisingly good agreement between the results of the simulation and the actual distribution of stars. And this agreement is surprisingly good given the simplicity of the model assumed by us. And the next slide will show a nice simulation. This is the simulations I was talking about, just presented in a nice way. We're having star formation episodes, star formation bursts at various times in the past and in various spiral arms. And then the stars, of course, change, they move away from their birthplaces. And the final image is then compared with the results with our observations. So in the final image, we'll compare the results of the simulation with the currently observed distribution. And so this indicates that indeed stars are formed in bursts in the spiral arms, but their current distribution doesn't trace the spiral structure anymore. And this is all I prepared. Thank you very much.
Thank you very, very much for this wonderful talk. You came ahead of time. Very good. Thank you. Do we have questions or comments to this lecture?
I do have a question.
Could you tell us who you are?
Krzysztof Meissner.
Ah, okay.
The question is why the distribution of Cepheids is so asymmetric on the map of the Milky Way, so that we have everything is to the south and there is almost nothing to the north or up or down in your map.
Yes, this is a very good question. So let me go back.
Exactly here.
Yes, but here is okay. So one reason is that we this is the sun, so that's where we are, and this is the galactic center with a black dot. So one reason here on this side, on the X and Y negative, sorry, X negative, Y positive, we do not see faraway Cepheids, which are fainter, because this is the northern hemisphere. So we could explain it with that. Here in this cone, on the other side of the galactic center, we do not see Cepheids because we are obscured by gas and dust. This is the only place in the galaxy where the gas and dust are so dense that even those Cepheids, which are so bright, do not manage to get through these clouds. So this can be an answer. But here we think that there are simply no Cepheids. So given that the Cepheids form in episodes, and we have a big clump of Cepheids here and none here, may suggest that these star-forming episodes were induced by some, but were triggered by something. For example, an interaction with a galaxy that was passing through, or
it looks like, yes, because
yes.
in your simulation, it looked arbitrary the time of birth, but it didn't, because at the end they are combined at the same in the same plot, in the same
yes
place,
yes
same
so because Cepheids are fairly short-lived, so these Cepheids are very you could imagine that if Cepheids if we compare the life of a galaxy to a 24-hour day, Cepheids are just the last half an hour. So there were Cepheids in the it is possible that there were Cepheids here and here in the past, of course, but the fact that we observe the majority at this moment in this place may suggest an episode of some interaction, in fact.
And what happened at the end of the life of a Cepheid? She stops pulsating, she's not
Yes.
very thin or whatever.
So being a Cepheid is just an episode in a life of a star of a certain mass and composition. So a normal regular star at some point of its life, given it has a sufficient mass, will become a Cepheid for some time, and then will stop pulsating and no longer be a Cepheid anymore.
That's a sudden process, this stopping, or not?
I don't know if I don't know if we have ever observed a stopping of pulsation, but I think in terms of the life of a star, yes, it is fairly sudden.
Okay, thank you.
All right, I don't see any more questions, but maybe still there is.
I have question. I have question.
Could you introduce yourself?
Marcin Gronowski from this department. I have two questions regarding to this star forming episode, because you have very nice demonstration that the star formation is not like a constant process, but it's rather like it happens in certain moment in the evolution of our galaxy, let's say this way. And I have a question, are you able to correlate this moment when there was a star formation, the most intense, with any of the process which happens in our galaxy? I don't know, some kind of a collision between clouds or collision with other galaxies. And the second question from the same line, do you think this kind of this way of formation is like a burst of star formation? Do you think it's only for Cepheids or their stars of given mass, or do you think it's like a general for any star of, I don't know, do you think our sun could also be formed together with many other suns at the same moment at time?
Okay, thank you. So let's start with the first question. So we astronomers think that there had been at about 250 million years ago, there had been an encounter with the galaxy, and we do see the Milky Way has many satellites, satellite galaxies that had interacted with the Milky Way in the past, and now they are just torn to pieces, if I may say so. We just see the remains of those galaxies. So yes, there had been interactions. In fact, there was we suspect that the interaction with the Sagittarius galaxy may have caused trigger intensive star formation. We do not know exactly how what episode could be responsible for this particular star forming episodes, but the Sagittarius galaxy is the suspect here. And then the second question, the Cepheids are just a small group of stars. There are billions of stars in our galaxy, and we could do it for Cepheids because just for Cepheids we can determine precise distances, but this should be true for any types of stars. So because Cepheid is a Cepheid just for a short time, and for the remaining time of it's just a regular star, and there is no reason why the star formation episodes would work for Cepheids and not for other stars. So yes, the star forming episodes can be attributed to all stars. Yes. I don't know. Does it answer the question, or maybe?
Yes. Thank you very much.
More comments or questions? I don't see on a chat. I don't hear anyone. So we thank you also very, very much, and we eagerly await a Nobel lecture as well.
Thank you.
Thank you both for fantastic lectures and for the prize. Congratulations. And thanks to the founder of the prize who made it possible. Thanks to everyone, to all the listeners, and we close this meeting. Thank you so much.
Thank you.
Bye.
Thank you.
Goodbye. Thank you.
Bye.
Thank you.
Indeed, we forgot. Merry
Yes.
Christmas and
Yes,
very
Merry Christmas
good
to everyone.
evening to everyone. Thanks for reminding us.